With our apparatus stated, let's go on to the main procedure. We place a pond with Elodea upside down in a test tube containing dilute sodium hydrogen carbonate solution. So over here we see that this test tube was not turned upside down this time. So here we have our seaweed with the cut end pointing upwards. Now this sodium hydrogen carbonate solution is to ensure that there's a constant supply of carbon dioxide. Now we place the test tube in a beaker containing fresh water, usually at 25 degrees Celsius. Okay, so we need fresh water at usually 25 degrees Celsius. But this time around, since we are going to do something with temperature, we are not going to do the usual 25 degrees Celsius. We are going to either increase or reduce the temperature. Then we darken the room. This is to ensure that the lamp will be our only source of light. We place the lamp at a fixed distance from the plant. Next, we are going to count the number of oxygen bubbles given off by the plant in a 1 minute period. This will show us how fast photosynthesis is taking place. If photosynthesis is taking place at a fast rate, more bubbles will be given off in a minute. If it is very slow, less bubbles will be given off in a minute. Now we repeat the experiment at different temperatures. So we ensure that we are going to have at 0 degrees, and then we vary the temperatures at constant intervals. Okay, so from 0 we could have 5 degrees, 10, 15, 20, and so on. Each temperature change is going to give us a varying number of bubbles released. Now the last thing we're going to do is we are going to graph the results, placing the temperature on the x-axis and then the number of bubbles released on the y-axis.